my name is Chuliang. I'm currently an assistant professor at UCLA, and I'm a theoretical ecologist. So this workshop has really brought a lot of thinking to me and has been super fun. And today we'll be talking about something that be typically larger than a genomic scale, but thinking about microbiome and together with the host. So here is a classical view of the speciation. So we have a ancestral lineage, and there's some isolation event happening. For example, a change in mountain range, and we have two lineages. And because of different natural pressures, one might evolve a green wing, another evolve a gold wing. So the gene flow reduces over time, and the reproductive isolation is enhanced. And this prosthetic paradigm basically really sees this reproductive isolation as being something typically fixed. So they're really hardwired into the genome. So in a sense that the speciation is usually considered permanent or irreversible evolution endpoint. Once you are speciated, there's no way of going back. So that's been the dominant view of speciation for the past decades. But it has recently been shown that this is really not the full picture of speciation, at least a major class of speciation do not play to this script. So to show you this case, let's visit a beautiful experiment done by Barker and Bolstein. So they worked with a Nasonia wasp, tiny wasps that are model system for speciations. So once they have this parent cross between the Nasonia vitropianus and Nasonia guruta, you can see that the survival is high, but their crossover offsprings have much lower survival rate, a classical reproductive barrier. However, once the biologists actually move the genes, basically make them gut microbiome free, then you actually do not have the reproductive isolation again. It's basically completely almost reversed. And once they actually reintroduce the gut microbiome, the reproductive barrier come back. So in other ways that with the gut microbiome, you have the reproductive barrier. Without the microbiome, you don't have the reproductive barrier. And this raises a fundamental question. If the speciation in this, at least in this system is reversible. So this raises a fundamental question. So how, if speciation are dependent on the microbiome present, how does this change our predictions and understanding of the dynamic of speciation? So to answer this question, we will need a mathematical model. The most classical canonical formalization of speciation is known as the BDM model. So for those of you who are not familiar with this, so we have this ancestral population, for example, with big A, big B, and the combination of genotype little a, little b would be lethal. And then with isolation event happening, each isolated population mutate. One mutates a little b, another mutates a little a. But since little a and little b are not present at the same time, they are not lethal. However, and over time, this new allele basically get fixed. But then once they actually get contacts again and hybridize, then the offspring would carry this new genotype, little a and little b, and this become lethal. So this is a classical example of how reproductive isolation can happen without either of these mutations being lethal during this process. And while this classical BDM model is like this, it's not the most easy way to work with mathematically. So here we could use a framework introduced by Sergey, basically reformalize this BDM model as a random walk on the wholly finished landscape. So what he did is essentially that we start from some ancestral state. For simplicity, let's just consider a haploid, little a and little b. And then with just one mutation, this little a, big B seems to be non-incapable to be an inviable whole in this field landscape. And the only viable intermediate state would be big A and little b. And eventually, once they reach the final state, with big A, big B, then this derived species would have this reproductive isolation with the ancestral state. And with this framework, we can easily use the mathematical ways to calculate its speciation times. So for example, we can consider here a, sorry, I can't make it. Oh, okay. Yeah. So for example, with two locus, there's only two passes going on. So each pass takes an expected time of 1 over mu, where mu is the mutation rate. And there are two states, so going to take 2 over mu times. But in contrast, with 4 locus, for example, we consider more complicated cases where there will be 2 incompatible rules with little a and big B and little c and big D, and we have more complicated landscape. But we can still do the mathematical calculation carefully, and we can calculate the expected time for speciation. And I just want to mention that this may diverge from the BDM model formulation in the beginning, since in the BDM model, initially you have 2 separated lineage, and here essentially we only have 1 lineage, and we're considering this the final state with the initial state. But it can be proven mathematically that they only differ by a factor of 2. So all the calculation will be much easier using the single lineage, just mathematically, and all the result can be translated back with the 2 lineage. So there's a mathematical equivalent in this sense. But then how do we actually encounter this effect of considering the microbiome and together modeling the Holobiont? The original authors of that science paper proposed their own model, where they basically call this microbiome-assisted BDM models. And in this framework, what they did essentially is that with more symbionts, there will be more incompatibilities, and the speciation must accelerate with the presence of microbiomes. However, from a theoretical perspective, this is not usually the most satisfying answer. One way is that it's really a combinatory argument. So it's really just because with microbiomes there will be more incompatibility, so always accelerates. And they do not really consider that mutation is typically considered irreversible in these processes. And actually, under some cases, as we will show later, that with the presence of microbiomes, sometimes actually slows things down. And so here we propose a new model where instead of specifically modeling the effect of the microbiome host genome incompatibility, we essentially consider the microbiome effect as essentially a mapping on shifting the epistatus landscape. So suppose that with one microbiome environment, the host gene incompatibility would be little a, big B. And then suppose we introduce another microbiome environment, and it has a different set of rules, like now the microbiome has essentially a changing the epistatus. So the new host incompatibility will be big A, little b. And then now we would have these 2 layers of the landscape. And with the change of the microbiome, essentially we're having the random walk along these 2 layers with the switching process happening. And we can calculate how long it, again, similar to the traditional surrogates approach in modeling the BDM model, we can also calculate the first passage time about how long it takes for speciation to happen. And mathematically, this will be modeled as a special class of the Markov process. So this model is exactly solvable for a large class of parameters. And here are the key results. So first, if we just focus on 2 locus, with 2 environments, we can only come up with 2 sets of incompatibilities like this. And we can prove mathematically under this scenario, the ability to allow it to switch would always slows the speciation time. So it always takes longer for speciation event to happen. So here, the x-axis is the environmental switching rate. Y-axis is basically the mean time to reproductive isolation. The static baseline is basically that if they're only in one static environment, how long it takes, so 2 over mu. But we can prove mathematically that if in this class of model, if we allow speciation to happen, it always takes 3 over mu. So it's always going to be a 50% penalty allowing to switch. So again, this shows that Barker and Bockstein's model does not capture the full possibility with the presence of the shifting landscape. But how about a more complicated cases? If we just go beyond this more simple 2 locus case, what happens? So here, if we allow the host loci to change, here the x-axis is basically a host loci. The y-axis is the acceleration ratio. So if the acceleration ratio is larger than 1, it means that it's faster to speciate. If it's below 1, then it's going to be the acceleration of the speciation time. Let's see what happens when we allow the epistatus incompatibility rules to be just between a pair of the locus. And this is what happens. When we have actually more than 10 locus, then it actually shifts from being the acceleration process to acceleration. So as the host genomic architecture become more and more complex, actually starts to have these acceleration factors. So in other words, the shortcuts really takes over beyond these traps introduced by these two layers. And if you allow a higher order epistasis, you can see that the acceleration becomes more of the rule instead of the exceptions. And with epistasis order 4, all of the cases will be acceleration. And so far, we have made this very strong assumption that basically that once the microbiome environment switch, the incompatibility rule basically randomly change. So here, essentially, we're at the... So to account for this, let's introduce the idea about some genetic correlation between the incompatible rules. So on the x-axis, it should be correlation between the rules. So if c equal to 1, it's basically the rules are identical, so nothing changed. If c equal to 0, it will be random rule. So what we have shown you before. And this is what happening. So for the cases, most of the cases are de-acceleration. So of course, when c equal to 1, it has to reach 1, since in this sense, two environments are basically identical. So there should be no change in the speciation time. So as you can see, once the initial effect with random correlation to be de-acceleration, it actually fast reach to null effect. But things completely changed when we have a high locus or more genetic complexity. As you can see, the acceleration factors basically holds robust across a larger range of the correlation regime. And this holds for larger number of loci. Another question is that, well, then would there actually be a limit to the acceleration factors? Does introducing a Holobiont always give us unlimited speed up to speciation? And what we found that actually no. Interesting that, so here the x-axis is basically the rule count, so how many rules we have. So y-axis is basically the limit acceleration factor when we allow the loci to go to infinite. So basically we use a random matrix approach to calculate the case when the loci number goes to infinity. And this is what, and the null expectation basically that there's been no acceleration. And this we found that the factor can be shown that it's always going to be upperly bounded between less than 1.7. So the acceleration cannot have a factor larger than 2 in a sense. And we can also do it for another case where, so this has been how the Alan Orr has been modifying this BDM model, where it has been introduced this snowball effect. So instead of the fixed set of rules, he basically let two pair of loci have a fixed probability of being incompatible. But also in his model, the maximum effect of this acceleration is fixed, still going to be less than 2. Then is there really no way that we can use a Holobiont to actually accelerate this to larger than 2, larger than a factor of 2? And this we found that if we actually allow the Holobiont diversity to change in a way that so far we have only considered 2 possible environments. But what happens if you have 3 microbiome environment, 4 microbiome environment? So here's x-axis is the number of the different microbiome environment. Y-axis is basically the acceleration factor. And this we found. So there should be no limit. In a sense that we can prove that mathematically that these acceleration factors would scale to the square root of the number of environment. So if you have 4 different environments, basically you get a factor of 4. If you have 9 different environments, you get a factor of 3. So to wrap up, I refine the idea that the speciation is not only hardwired to the genomes. The microbiome play a strong factor in actually determining many of these speciation factors, and it can make the speciation even reversible under some cases. Second, under simple genetic architecture, we can prove that the microbiome switching basically always de-acceleration the speciation. But with increasing genetic complexity architecture, it actually can most of the time accelerate this process. And third, we basically found that the key factor in acceleration would basically be the number of possible microbiome environments, which I think would be interesting since that the human, in many of the mammals, the gut microbiomes are not always static through their lifetime. And fourth, this is also something that really this workshop has brought me a lot of thinking. Since I started this project working with a bunch of evolutionary biologists really believe in holobionts, thinking of the microbiome and the host are basically the same unit of selection. But now I'm wondering, since actually the presence of microbiomes, epistasis, seems to actually speed up the speciation, which might be actually bad for the host fitness overall. So are they actually in conflict or internal conflict or not? I still haven't wrapped up my head around it. In the end, I'd like to thank my amazing collaborators. So this work has been led by my master student, Han Yang, who is going to go to Michigan this year to study his PhD. And also to acknowledge the founding source, as well as John Rothgarden, who has really shown me a real thinker about evolution biology. And thank you very much. Questions?
I have 2 microphones today. Doesn't make me any faster though.
Maybe I missed it, and if so, sorry. But would you say that the way that you've structured your holobiont switching would be just equivalent to sort of any type of environmental change whatsoever, or is there something about the way you've structured your holobiont to be a little bit different than just some sort of stochastic environmental change?
That's an excellent question. Sorry, I haven't clarified this. So in this project so far, we have not considered anything actually specific to the microbiome communities. So in a way, it's going to be environment. And we're thinking about some follow-up collaborations with SAS, which will be considering, well, essentially that if we have more than one holobiont, then there will be some horizontal transfer of this. And and this is still something in progress.
So just 2 quick clarification questions. So how are you defining the time to speciation? Is it the first point in time?
It would be the first passage time.
So it's the first point in time at which you're in an environment and at a genotype that in that environment is incompatible with the ancestral genotype?
Yes. So for example, here, if you are in the environment 1 and you reach this point, then this will be the time when you have reached the reproductive isolation. But suppose now you are in the environment 2 and you have to reach this point for the reproductive isolation time.
And then what are you assuming about the time scale of environment switching relative?
Yeah, that's a great question. So mathematically, we can do it for any. So essentially, the S factor here basically consider the time scale between the 2 processes. And for most of the theoretical analysis, we basically consider the fastest switching period. So essentially, the S goes to 0 for analytical tractability. But for some simple cases, we can consider the case for arbitrary S. So for example, for the case for 2 locus, we can consider arbitrary scale of the environmental switching rate. So you can see the S divided by mu is basically the relative rate of the speciation, the relative rate of switching divided by the mutation rate.